We are the paradoxical ape. Bipedal, naked, large-brained, long the master of fire, tools, and language, but still trying to understand ourselves. Aware that death is inevitable, yet filled with optimism. We grow up slowly. We hand down knowledge. We empathize and deceive. We shape the future from our shared understanding of the past. CARTA brings together experts from diverse disciplines to exchange insights on who we are and how we got here. An exploration made possible by the generosity of humans like you.
Thank you, everyone, for wonderful presentations. My co-chair, Jerry, and I would like to welcome everyone in the Q&A section. We have several sections from the questions from the public that we want to go over next. Okay, the first question we have is for Pascal Gagnon. And the question is, what is the evidence that we lived in small-scale societies for most of our history? And to what extent did this limit our ability to accumulate and spread knowledge?
Thank you very much for this interesting question. I would say the evidence is twofold. One is the archaeology, that with no evidence for even small settlements older than 20,000 years, and no evidence for domesticated plants or animal species until about 12,000 years ago. And then the second evidence is from present day foraging people who are natural fertility populations that live completely off the land, where birth intervals remain more than 3 years apart, a very, very high infant mortality. So there simply was no way to maintain population numbers as you then start seeing after agriculture in the last 12,000 years. So that would leave small groups. Now, how big these social networks really were, that is another question. And that relates to the second part of the question, the spread of knowledge. Even relatively small group that live today in places like the Kalahari, they keep in touch with people 200 kilometers away. And they tell stories about people who live that far. So there might actually have been, for these formative hundreds of thousands of years of small-scale egalitarian societies, an impressive degree of exchange of information and knowledge that might actually have contributed to these cognitive capacities that do not require schools and the teaching of math and everything we bore our kids in school with. I'll leave it at that.
Thanks, Pascal. Our second question is for Robert Clodender. Robert, a tremendous talk. The question is, what is the difference between center embedding and just mere symmetry? Because there's lots of symmetries in nature, both in body form and in things like termite construction and so on. How is that distinctive from center embedding?
Yeah, that's a really good question. So the way you would define recursion is that for any given pair of elements, the second one has to call back to the first one. It has to refer to it in some way. So you can think of it in terms of a computer program, in terms of bracketing. And in fact, one of the most recent studies that just came out at the beginning of the month actually replicated an earlier study from 2 years ago that actually worked literally with brackets of various types, curly brackets, parentheses, square brackets, angle brackets, and so on. The most recent study was done with crows, and it was found that crows were able to do the matching. So there has to be a dependent relationship between the prior element and then the recurring element later. actually, Stahl, when he first proposed this based on his study of Vedic ritual, it wasn't just symmetry that he was proposing. It was based on actual identity, well, partial identity relationships, at least between different rites and sub-rites that occurred in different portions of the sequence in this kind of symmetrical fashion. But the later occurrence would call back to the earlier one. And that's sort of
really helpful. You know, I had the same question myself. So it's re-entrant symmetries is the way to think about it. That's the recursion factor. And it's not just layers of symmetries. Thank you so much.
Our next question is for Evan. Among the about 1,500 genes that have undergone structural changes, do you see genes involved in neuropsychiatric disorders other than autism, such as schizophrenia, depressive disorders, etc.? Do you see any implications for the evolution of the neuropsychiatric disorders in humans compared to non-human primates?
I mean, there is an enrichment, at least for the segmental duplications, for genes associated with neurodevelopment. Interestingly enough, there isn't actually a strong association or enrichment across the neuropsychiatric disorders. So it's not as if we see a general one-on-one relationship that the genes that have structurally changed between chimps and humans are also the genes that are responsible for neuropsychiatric disorders and autism in humans. What I will point out, and I think it's relevant to the question, is that many of the duplications that I talked about that involve essentially the birth of new genes that are specific to humans are actually creating susceptibilities to rearrangements that are associated with autism, schizophrenia, and neuropsychiatric disorders more broadly. So, for example, one of the gene families that's expanded in chromosome 16 is one that actually promotes one of the most frequently associated CNVs that's found in association with schizophrenia and bipolar disorder. The same holds for autism. One of the gene families that's expanded very close to the centromere is a gene family that creates essentially the second most common form of autism in the human species. And what I think is most interesting is that architecture, based on our reconstructions of the Neanderthal genome, does not exist. And so this is a very recent expansion within the human lineage and the common ancestor leading to all modern day humans.
Thank you, Evan.
Great. Next question's for Anne Stone. So Anne, in your talk, you made the point that most genotype-phenotype correlation data is carried out on European or populations of European descent. And the question is, why is this the case, and how can we remedy the situation so that the data set becomes more representative going forward?
I think it's really because of the focus of biomedical research in European populations, and in particular, to really get at complex phenotypes, you have to have enormous population sizes. And so countries that have socialized medicine and large databases, and then have put forward the funds to really genome sequence a large number of, a large percentage of the population, have really given us the best data that inform us about these phenotypes. Now, as an anthropologist, sometimes the phenotypes that I'm interested in are not necessarily represented fully, right? Since these are biomedical, but they are our best sources of data, really. That is beginning to change. We do have projects that are focusing on people of other ancestries around the world, but I think it'll take the genome data is better represented, although there's still issues there. It'll take a while for the phenotype data to catch up.
Thanks so much. Appreciate it.
Sarah, the question is for you. If the majority of variations, about 85%, are within populations rather than between populations of about 15%, why are humans classified by race using the small between population differences?
And the short answer is they shouldn't be, because race doesn't correlate with genetic variation, at least race as it's defined in the US. So historically, race has been defined based on a combination of social sometimes behavioral characteristics, strongly influenced by the culture at the time, and also biological characteristics, things like skin color, for example. We know that's a terrible way to classify people. It doesn't correlate with so-called races because it's simply an adaptation to different UV environments. But historically, again, race was used to justify often hierarchical stratification and really horrible abuses that occurred. And so it really is a social construct, doesn't correlate at all, or doesn't correlate well, I should say, with patterns of genetic variation. And I'll say, just as an example, there was a classic study done by Noah Rosenberg and colleagues where they looked at global populations and they used something called structure to infer genetic ancestry. And it looked like the genetically inferred ancestry correlated in a way that may correspond with so-called races in the US. But when I, for example, expanded that study to include many, many more Africans, and then other people expanded to include many, many more non-Africans from very diverse populations, that broke down. We no longer saw this classification by continents or by so-called race. In fact, there can be more variation between populations in Africa than we see globally.
Yeah, thanks, Sarah. Next question is for Dan Geschwin. Dan, it's a kind of complicated question, but let me walk through it. Of course, you talked about how human brains have expanded not just in size, but in complexity at the level of neuronal connectivity and at the molecular level, as you explained. And the question has to do with the way in which this growth occurred evolutionarily. And, so how did this expansion occur in a sort of progressive fashion evolutionarily? And then here's the kind of provocative part of the question. And does this say anything about the regions that are most susceptible to neurodegeneration, to decay through aging and other processes?
I focused, and I think we know most about potential, models and mechanisms of cortical expansion based on, on neurodevelopment. And so, and in fact, most of the kind of size and connectivity expansion that has to do with the expansion of the layers 2 and 3, the superficial layers that connect across the two hemispheres and long-range parts of the cortex, involved expansion of progenitors and progenitor pools in this outer subventricular zone, most likely. And, there's a lot of evidence for that. And I kind of focused on that since that's something that we understand mechanistically. And I think the message there is that part of the regulation of those genes, which are highly constrained, this, I think Evan, pointed out somewhat in his talk, we're talking about very constrained developmental genes that can predispose to neurodevelopmental disorders. They're also the areas that regulate them. It's kind of almost it's not really a paradox, but the fact is that the protein coding sequences of those genes are under a lot of constraint, and yet the regulatory regions seem to be what have evolved the most rapidly. So that's what I kind of focused on. But I think that, there are many, many mechanisms. And in fact, the mechanisms of the molecular adaptation and expansions aren't really that well understood at all. And it's probably a number of different, it has to be a number of different mechanisms, from the kind of structural variation that Evan talked about to changes in regulatory variation.
Thanks, Dan. Is it reasonable to hypothesize that those are last to evolve or the first to generate? And if it's more the regulatory networks, that's where some of the challenges are in neurodegenerative disorders?
Well, if you actually partition the heritability that we have now in the current state of neurodegenerative disorders across the genome, they don't seem to coalesce into these very, very constrained, human evolved elements like the HARS or HGEs. It's the developmental disorders that do. So I think it's something else, something fundamentally different. And one of the things we have to recognize is that, we can put mutations that cause many human neurodegenerative diseases into mice, and they get a phenotype that's extremely similar. So It's not every case, but many, many of them. So a lot of the mechanisms of neurodegeneration are likely to be very conserved. Not all, but many, and involve kind of basic cellular processes such as protein homeostasis, et cetera.
Very good. Thanks so much.
Carol, the next question is for you. Are genes that are controlled by GATA3 transcriptional activity also controlled by other transcription factors? And how might genes that are controlled by multiple transcription factors be affected across different types of primates?
That's what I think a lot of people are studying that. So there's a lot of unanswered questions as to how many transcription factors, what happens when multiple transcription factors during evolution or during control different genes in different species. With regards to GATA3, GATA3 is part of a family of transcription factors that have a specific binding site, and there are other members of the family, GATA1 and GATA2, that are also known to bind to very, very similar sequences. With regard to our experiments, we didn't see expression of GATA1 or 2, so we don't really know what these genes are responsible for doing, at least in the brain cells that we generated from humans and non-human primates, because the expression of those genes, so those genes are present in the genome, but they're not being expressed at levels that were detectable. But there might be others. There might be other genes that are binding to similar sequences as GATA3 does, and that's something we will further investigate.
Thank you.
Okay, next question is for Terry Sanowski. Terry, I love this question. I think you're going to know why. So you talked a lot about the large language models and their ability to self-train. And the question has to do with, has anyone begun to take the next step where those large language models evolve in a Darwinian sense? You know, there was this evolutionary programming branch of AI and then machine learning branch 1, but can machine learning start becoming Darwinian and, you know, in an actual Darwinian sense of population dynamics?
You know, that's a fascinating possibility. And, you know, there have been people who have not within large language models, but within the larger field of neural networks have developed these models, which require a different context, a different way of being built. Right now, these networks are built. You pick the architecture, of course, that evolves in animals, but then you have experiences, which means you have data, you're learning something, but they don't have a developmental period, right? They don't develop. And that's really what you'd like if you want to evolve something. I think it's a lot of that are developmental genes that were discussed earlier. And there are a few researchers out there who are actually incorporating development into the construction of these networks in the context of locomotion, of how to evolve a creature that can crawl toward a goal. And so, yes, the answer is that there is a branch of machine learning that is incorporating development and evolution, but it's not mainline yet. But I think it's going to be an important component. I think that in the future, AI will have to develop, you know, have a period of development, just like the way that we have a long childhood.
Very good. Thanks so much.
Johannes, the question for you is the following. You highlighted the importance of finding new fossil sites. Many fossils have been found in the East African Rift Valley, which offers favorable conditions for preserving and finding fossils. What other African regions do you think will become important for finding hominid fossils in the future? And what about Central and West Africa?
So yes, what we have currently is probably limited to South Africa and Eastern Africa, but there are also sites in Western Africa. There are also sites in Northern Africa. So it's about like, how many of those sites have we found so far? Otherwise, early hominins have been living in a lot of places within Africa as early as like 300,000 years ago, or even before. So their distribution must have been much wider. It's just that those sites where this early hominids may have lived are not exposed. The sediments are not exposed, and that's why we're not finding them. Otherwise, we have some evidence from North Africa, some from West Africa. So I would say that they must have occupied most of Africa. And if the right sediments were exposed, we would have found some hominins. But it's just the geological phenomenon has not exposed a lot of ancient sediments in most parts of Africa.
Thank you.
Okay, a somewhat provocative question for Pascal. So Pascal, you talked about how both biology and culture have shaped the course of human evolution. Of course, we now live in the techno-scene as well as the branch of the Anthropocene. So how is technology, and in particular medical advances, where we're changing the actual hardware that runs the software that is human culture, how do you think now the intersection of biology, culture, and technology is going to shape evolution going forward? When the singularity comes, will evolution stop or accelerate? I've added that part to the question.
Very provocative and a huge question. I might start with a joke that we might hop into the evolving learning machines, and then you don't need the biology anymore, which begs the question, who builds the machines? But I think that there's been a concern for, ever since Western medicine existed and people started thinking about the genetics of, allowing people to survive and reproduce, that would be very unlikely to do so without medical intervention. There, the immediate answer is that the vast majority of humans alive on the planet, don't see doctors, have no access to a lot of that advanced biomedicine. So you might even go as far as saying that the genetic insurance for humanity is with the populations that are not changing the effect of deleterious mutations. These effects are still, they curtail survival and reproduction. This being said, clearly in the more opulent parts of the world where people have enough funds, where there is a technology that is accessible to many people, many of us hope to live much, much longer lives and not succumb to, endless numbers of diseases that have killed our ancestors in absence of such medical advances. I'd like to point out, though, that the connection between technology and biology, we've heard from Dan Geschwind and from Evan Eichler how the genetic architecture of our intellects and our minds seem to be really, really fragile. Right? There is countless ways before you even born, prenatally, where biological variation in a period of time that's almost untreatable. Right? Who is drugging pregnant mothers? It's not really happening. It's just so risky. And there the biology plays out. So I think we're very far from uncoupling our biology from technology. And I think the dream of using technology to escape the biological constraints has happened to some effects, especially for rich societies, but we're very far from it. So that would be my attempt at answering this really, really good question.
I'm relieved. We can at least sleep tonight. So thank you.
I was about to say the same thing. Phew. All right. Robert, the next question is for you. Spoken language seems to have an important difference compared to written language. In spoken language, you cannot refer back to a previous part of the sentence unless committed to memory. Do you have comments on the difference between center embeddings in linear domains like spoken language and domains that allow for random access like writing, visual arts, and so on?
I oversimplified both things just for lack of time, but on the spoken side, there was a really interesting occurrence in last week's Saturday Night Live Cold Open, where Mikey Day had to read just one level of embedding off the teleprompter and clearly had to rehearse it because it was every candidate Trump-backed lost last week. And if you think about it, it's fairly straightforward, but just because of the center embedding, he really struggled with it, clearly, in rehearsal at least. And it's sort of interesting. You could say, oh, well, it could be from a phonological perspective, it could be the fact of having three primary heavy stresses on monosyllabic words, Trump, back, lost. It starts to sound like a list of words and the syntactic structure just disappears. So we could say it's that. It doesn't seem like that's a working memory problem because things aren't that far removed in space or time. But we can fix it in various ways that don't have anything to do with either working memory or time. So for example, if they had just put in a complementizer, that, as we call it, every candidate that Trump backed lost last week, all of a sudden the syntactic structure is like back again. And there's other ways of fixing it as well. So that's one side. On the written side, it is true that there's more center embedding in language that's written than in spoken language. But even there, you can start to see that languages have come up with devices to sort of massage the structure to lessen the burden, even in writing and reading, where you can look back and backtrack and so forth. And that includes use of, for example, subject relative clauses rather than object relative clauses at the most deeply embedded level, using sort of generic head nouns for those relative clauses like people, persons, things, someone, things like that. And there are other devices that are used in writing as well that seem to lessen the burden. So it's not just working memory and it's not just the time. There's more stuff going on, and that's currently under investigation in part by my graduate student, Emily Davis.
Great, thanks. Yes, next question's for Evan Eichler. So Evan, you talked about the duplicated regions and their relations to the development of the human brain. And of course, very appropriately, this is an anthropogeny symposium, but the question has to do with other effects of the duplicated regions not related to the adaptations of intelligence in the brain. In particular, the fight against pathogens, right, which is what did our ancestors in. Or I would add to that, the fight against trauma, right? So when I look at BOL A2B and iron homeostasis, maybe that's all about just hemorrhage and not so much about the brain. So, how do you kind of balance the driving forces here, those that are cognitive related versus those that are just surviving the pathogen and surviving the gash in the leg?
Yeah, for sure. I mean, let me just start by saying that I'm both anthropocentric as well as probably neurodevelopmental centric in my perspective, right? But having said that, when we look at the genome, it's an unbiased view. And I will say that, and it's been known, for at least 25, 30 years, that the immune response genes are some of the most structurally variant regions in our genome. The one thing I would say to that is that they're structurally variant in almost every genome that we've looked at, and we've looked at many outside of humans. So they show a lot of variation. And obviously every mammal has to survive pathogens. And so it makes sense we have that. I think, there are some really interesting things that we saw when we started looking at duplication patterns in chimpanzee. In fact, some of the strongest signal comes from immune response genes as opposed to neurodevelopment in the chimpanzee for chimpanzee specific. And it's not just gains, it's also losses. So there's some observations that were made even when the genome was sequenced that there are interleukin genes that are completely lost in chimpanzee and bonobo that are present in all other apes. So there's some really specific events that have happened to actually change the immune repertoire in each of our genomes. The other thing I will say to that is also there's really remarkable examples that go back now 50 years almost in terms of really unusual patterns of selection that are very deep, like balancing selection over the HLA locus. And that involves both diversity in terms of the single nucleotide variation, but also complex structural variation events. So for example, when we were sequencing the macaque genome, one of the things that came up when we started to sequence the MHC regions is we started to see very large expansions of genes that are not expanded in any other primate, which begs the question, when people think of these primates as interchangeable models in study of research, in particular in terms of inflammation and immune response genes, what does a much larger, more complex set of genes in the MHC regions in the macaque do for that as a model with respect to human disease? But yes, short answer, lots of variation, specifically in immune response genes, and there's even huge amounts of variation between individuals in the human population.
Very good, thank you.
And the question is as follows. Your research involves understanding how diseases such tuberculosis emerge and evolve. Could you please expand on how ancient DNA research on diseases could inform us on how we prepare and respond to global health crises, such as COVID-19? And how can this knowledge be applied to global health policy?
science gives you information, policy is what you decide to do about it. And often those two don't seem very closely related. But I think, when you use ancient DNA to study pathogens, and you add that time dimension, I think you very quickly develop a One Health perspective. So you think about how the pathogens move not just among human populations, but among us and other species. So tuberculosis is caused by the Mycobacterium tuberculosis complex, a group of, it's basically the same thing, but they have different names if it's in a different species. But any of them, if they infect you, you're diagnosed with tuberculosis and you're treated the same way. And what we see from ancient DNA, like from many other pathogens, is a history of exchange back and forth among species and movement of pathogens with our trade routes, but also with interactions with other species. You also quickly see how much the increased density, population density with agriculture has influenced pathogen patterning and exchange. And so tuberculosis was thought to be a really ancient pathogen, but all of our genomic data now suggest that it probably jumped into humans in the last 6,000 years. So since agriculture and not before. But there's a lot of areas where we need to know more, and we can also get a good sense of what the variation in some of these pathogens was like prior to vaccination and prior to antibiotics, and those can give us insights into the biology of the pathogen and potentially treatments. But how that's going to impact policy, one would hope it leads to greater support for surveillance, but we'll see.
Thank you, Anne.
Next question's for Sarah Tishkoff. Sarah, I actually had the same kind of question brewing in my mind myself, because the medical literature is confused about the effect of increased activity of alcohol dehydrogenase, right? Because more ADH activity means more acetaldehyde, and that means pain, right? That means hangover, that feels bad. So is the effect of having increased ADH to prevent you from drinking too much alcohol because you feel the pain, or is it literally just you're clearing alcohol, you're lowering your blood alcohol level, and so you can actually ingest and metabolize more? So now let me get to the specific question, because I think it's really a fascinating one. So thank you, questioner, for this. And this is, I've never heard of this before, but tej, I guess is pronounced, or tej, Tej, is a traditional Ethiopian drink made from fermented honey. And the question is, could the resistance to alcoholism in Ethiopians be related to the drinking of tej? So it's now that general question, but now tied to a specific cultural question. So please, thank you.
Right, that's a great question. We asked ourselves exactly the same thing. That was originally what came to our mind, was this drinking of these fermented beverages and even foods that are common in that region. And the truth is, we really don't know. I mean, people have speculated a lot, but we really don't know what the selective force is. But what I will say is that in my entire career, I don't think I have ever observed as strong a correlation between let's say a haplotype. It's really a haplotype that shows this signature of selection, and the practice of some subsistence practice, right? In this case, it's agriculture, or a correlation with diet, let's say. As we observe at this locus, and the only other example that I can think of, it has to do with lactose tolerance. So mutations that regulate the expression of the gene lactase. I was frankly really shocked when we saw this, and what a strong correlation there was with diet, even when you control for groups that have the same genetic ancestry. Whatever it is, it's a really strong selective force. It's somehow correlated to agricultural diet. But as I mentioned in my talk, there is very strong selection. You see this extended haplotype And there are many different variants in genes that have important roles, often pleiotropic roles. They can affect different traits. One of the genes that also has a variant showing a signature of selection can influence lipid metabolism, for example. And it could be, therefore, that there could have been selection on one of these neighboring genes and this got dragged along. It could be a combination of these different variants on the same haplotype background, which is frankly what I think is probably very likely. And so as we begin to know more about, people do more genotype phenotype studies, and you figure out what is the impact of these different variants and these different genes on phenotype, we're going to learn more. But at this point, it still remains a really interesting question.
So stay tuned. Yeah, thank you, Sarah.
Then the question is, you mentioned that human accelerated regions are genetic regions that are changing faster on the human lineage compared to other primates. Because of the rapidity of change, are these regions more susceptible to maladaptive outcomes?
If I understand that correctly, one's asking, is it kind of like Evan's thing where these regions actually become more susceptible, around these human duplications or structural variations? And I don't think that is really known. I certainly don't know that, but I want to clarify one thing around that. We find that those regions are regulating, at least at a first approximation to the best we can tell globally, genes that are under a lot of constraint, that are expressed very early during fetal development, and that are really conserved, across mammals. And so the notion is that the genes themselves are going to be very susceptible to loss of function mutations in them, and they'll also be susceptible to changes in their expression as well, but that has less of an impact. Another thing that kind of happens is as genes, a lot of these neurodevelopmental genes, the average brain gene, I think, has about 6 enhancers or so, maybe a few more, it depends how you want to assign them. But and these are regions that positively regulate those genes. A lot of these developmental genes have more. And so the point is, there's a lot of redundancy in the regulatory, and maybe not redundancy is not the right words because we don't really know, but each gene has many regulatory regions. And so that a slight change in one of those regulatory regions might affect a phenotype, but it's not the same as a change in the sequence of that gene, which will be expressed right everywhere that gene is expressed in every context, whereas a change in a regulatory region may have a more subtle effect at a particular time in a particular cell type. And so it actually gives you, one could think a little bit more buffering from that perspective.
Great, thank you.
All right, our next question is for Carol Carmanchetto. So human and ape brain, other primate brains develop similarly, but as you so clearly show, it's just beautiful data with the iPSCs. It's the tortoise and the hare, right? So it's much slower to develop the level of neuronal activity in the human iPSC-derived neuronal cells, but of course they reach a higher level of activity, and that's just the general pattern. The question is, how do you explain how this evolved? How this difference in pacing evolved? And why is this difference in pacing a benefit to us? Now, you gave a specific example about GATA3 transcriptional regulation, but how did this all come about in a stepwise selective advantage manner?
There is a lot of research on that, and the actual evolutionary origins of human neoteny are still a topic of a lot of discussion. So we know that neoteny in humans is the slowing, the definition is the slowing or delaying of development compared to non-human primates. And that results in the retention of some of the juvenile characteristics. So some of the features related to neoteny in humans compared to non-human primates are physical features, such as we have a large head, a flat face, relative short arms. We look juveniles compared to the other non-human primates. So some suggest that those characteristics could be bringing some advantage because we were not perceived as aggressive or as challenging to to other individuals that didn't have these characteristics. But there is a lot of this very speculative discussion.
Very interesting. Thank you.
Harry, here's a question for you. There are a lot of arguments for and against claims that large language models like Lambda and GPT-3 are sentient. What capabilities or characteristics, in your view, are critically missing, which, if addressed, would perhaps allow a future AI to be rightfully deemed sentient?
That is a topic which is under great debate right now, and I've written, by the way, a archive paper specifically on that topic. So if you're interested, just Google archive and Sanovsky and it will pop up. But very briefly, the problem is that we can't agree amongst in the field a proper definition for sentience. Or for that matter, people argue, do these large language models understand what they're saying, right? Or are they intelligent? Again, those words mean, the different things to different people. Consciousness. There's no scientific definition of consciousness. There are many, many properties that you can assign, but there's no real way of measuring it. Well, except when you obviously lose consciousness, but and really here's the problem. The problem is if you take any of these words, sentience, you look it up in a dictionary, what do you see? A bunch of other words. You look those words up, it's all circular. It's not based on any fundamental scientific basis that is and for me, what a scientific basis would be something where you can tie it to ground it in specific maybe neural circuits, or maybe in even molecular signaling pathways or something, and ultimately we'll probably have as a mathematical definition for all of these words. But right now it really illustrates, I think, how little we really understand about these words that go back to the 19th century psychologists. We've been using them as if they have a clear meaning, but when you have something like a large language model, it really challenges us to come up with some deeper basis for which we can judge whether these words. And it's again, I think that we rather than argue about it, which we like to do, it'd be much better to focus on trying to really understand how these large language models are accomplishing, what's going on inside that allows them to respond and talk and to at least give some semblance of what you consider human sentience. But I don't think it's human, right? It's not a human, it's some other kind of sentience. Maybe it's just, I mean, by the way, the same argument comes up, are animals, are they conscious, right? Do they have same consciousness as humans, or what kind of consciousness do they have? I mean, a lot of people argue about that, and different types of intelligence. Again, this is something that we need to really explore in greater depth.
Thank you, Terry.
Next question is for Johannes. So Johannes, in your talk, you strongly made the argument that we need to get back in the field, and your own work and incredible breakthroughs come from being in the field. The question has to do with what new technologies are coming down the pike to help us get more information in the field, finding, extracting, and analyzing fossils. You talked toward the end of your talk, you mentioned geospatial science and using those technologies to help us know where to go and how to proceed. Can you get a little more specific? What technologies do you see coming online that are going to make the difference in the field?
When I talk about geospatial innovations and new discoveries in those areas, the way they can help us in the field is actually by giving us better opportunities to locate new sites. Because in history, if we go back in time to like 70s and 80s, most of the survey and exploration was actually done on ground truth check, which sometimes you end up in areas where there are no fossils and so forth. But now even Google Earth could actually lead us into like, good places where we can actually do field survey and exploration. So when I say new technologies in geospatial sciences, it has to do with like the way we document our discoveries in the field, especially in like, locating where we're finding those fossils, and, also identifying like possible what could be possibly fossiliferous sites from like, non-fossiliferous sites that are just volcanics and so forth. So we now have better approach in terms of how we can distinguish different lithological aspects, in the field. So it kind of reduces the amount of time and energy we spend to actually locate sites, and that's one advantage that we have. But in terms of like the documentation is something that has been revolutionized because of all this new technology that we have in QGIS and other methodologies of like stratigraphic identifications and so forth. So those are what are helping us in terms of like identifying new places. But what I really wanted to emphasize was that because of all these technological advances we've made in the last 2 decades or 3 decades, we've been able to locate new sites. But again, the problem now is how do we get there, and how do we survey and explore these new sites? And what I really wanted to emphasize was like, we're now not looking too much into like new discoveries in the field, and because it's really expensive, right? But what I'm trying to say is like, unless we do survey and exploration, make new discoveries, we're not going to still fully understand our evolutionary history. I mean, and Anne mentioned about ancient DNA, right? And we do need additional ancient DNA data. So where do we get those new data, right? We need to make new discoveries. And I think all of these new discoveries in geospatial sciences is actually helping us find sites much easier than we were able to about 30 years ago. So those advances are something that are really encouraged, but then the ground truth is always going to be like part of that fieldwork that we need to keep supporting. That was the point that I was trying to make.
Thank you, Johannes. Can I ask just a quick follow-up question to push a little bit more on the technology? So you pointed to precision mapping, obviously is very helpful. But what about new discovery technologies, things like hyperspectral imaging, things like ground penetrating radar, sort of force multipliers for the individuals in the field? What do you see coming in as far as technologies for discovery?
Well, I believe there are some technologies already, because there has been some technology to actually locate some ancient bones, like in Asia, I believe, a long time ago. But we haven't used that technology in any of the paleoanthropological sites in Africa. But honestly, would it be really fun to actually use those and find fossils than actually going out there and finding the fossils yourself? That's a different story. But again, that technology doesn't exist yet for the kind of work that we do, but I wouldn't be surprised if those kinds of technologies were developed and we use them in the field to find fossils. The only problem is, even if you have like such an instrument to identify fossils buried underground, would they still be able to distinguish between what is a human ancestor fossil versus, a gazelle or things like that? And I don't think we're that close to that kind of technology yet, but it would be great to have those kinds of technology.
Yeah, fair enough. I mean, then machine learning on the back end, but then, yeah, that's a lot to come together. All right, thank you so much.
Pascal, could you expand on anthropogeny's relevance to today's society? And do you see a place for philosophers, specifically philosophers of mind, language, or science in anthropogeny?
Huge question. Do I have half an hour, or how much time do I have? So I start with the end of the sentence. Yes, I think all science is philosophy, and we need good philosophers. I think one of the problem with our, historically, the understanding of how humans tick had to do with, ill-informed philosophers who, like Hume, would say that we're all selfish brutes who lived short, miserable lives, or like Rousseau tried to believe in some wonderful past where everybody was fantastic. All you have to do is make a wonderful, just society, and then everything is hunky-dory. So I think one of the big promises of people who do comparative psychology, studying behavior of non-human primates, other highly social animals, and study different human societies, is that we come to understand that humans are both incredibly selfish and incredibly altruistic, that we have these astonishing capacities to cooperate, not just in groups, but in groups of groups. But the downside is we often do so in order to compete with other groups of groups. So I think that one of the promises of anthropogeny is that it might provide some slightly more realistic insight in what humans are, in the worst and the best. And boy, do we need that to address, global challenges that we face right now. And so I think we won't be able to do that without philosophers of all these specializations.
Thank you.
Thanks, Pascal. Next question's for Robert Klunder. So Robert, you talked about broader application of linguistics. And the question is, can you tell me more? What can you say? What questions do you have in mind where linguistics could be applied more broadly? What sort of applications specifically are you thinking about?
I think there's like a good story to be told for the evolution of arbitrary symbolic reference that could be pulled out of animal ritual. That actually was proposed long ago by Robert Brandon and Norbert Hornstein in biology and philosophy in like 1986, sort of referring back to Edmund Wilson and I think Lorentz also, where you can see how animal behaviors start to move away from their home domain where they're actually functional, where they start to get where they're sort of transported into a different behavioral domain, particularly in courtship domains where the behavior is no longer functional. And so it starts to move away from its original mapping onto a real world function. So that's one thing that I think could be investigated. Another one is, and this goes back actually to Panini, who was probably the first one to talk about this, what we call in linguistics semantic roles. And that has to do with the argument structure of a predicate. So, what's the agent or the actor? What's the undergoer or the patient? What's the recipient, the instrument, source, goal, and so forth. And then the question is, how do those things map onto syntactic positions, which is called the linking problem in linguistics. And that's actually a fraught proposition, because as you can see in a simple sentence such as, I gave Pascal the book, Pascal is the direct object, but if I say I gave the book to Pascal, now it's the object of a preposition. And there are a variety of alternations of predicates like that across languages. And so that problem is difficult. And I have my suspicions, and this is one of my crazier ideas, that we can actually again look to animal ritual, possibly for sources for that, because particularly in what I will call, for lack of a better term, sociosexual relationships, particularly in primates, these are negotiated interactions that are ritualized, they're not functional. And there's a basic decision of like who's doing what to whom in this thing. And there are a variety of things that go into it. It's not just dominance hierarchy determined, rather there are also issues of coalitions, status, with baboons in particular, whether they're harem leaders or not, or whether they're older or younger. So all of these things go into this calculation of like who's going to take the active role, who's going to take the passive role. So I think there's some sort of precursor for the kinds of problems that we see in linguistics in the linking problem. And in general, just point out that, a lot of what we do with language is pretty ritualized behavior in the first place.
Yeah, very thought-provoking, because you made the point in your talk that language is the suggestion it's an acceptation of human ritual, but from animal ritual, what other acceptations arise? It's very fascinating. Thank you.
Evan, are there any known examples of increased copy number variation that caused the reproductive or fitness advantage?
Yes, I was thinking about this, and probably the best example of this is on the Y chromosome. About half of the genes on the Y chromosome, and there's not many of them, are part of gene families that have expanded to high levels. And we know, based on infertility studies in men, that if you have reductions or deletions of those gene families, you actually have infertility issues. I think maybe in a more broader sense, you can think of examples like increase in the amylase locus, which allowed us to adapt to starch-rich diets. That's something that's been reported decades ago, and it seems to hold. This is an example of a human increase Even the BOLA2 example that I talked about in my talk, that's an example where we actually were asked by reviewers to estimate what the selection fitness would have been, knowing that that actually predisposes to microdeletions associated with autism. And it's an order of magnitude greater than what you'd expect for neutral. So there's good examples of increased copy associated with increased fitness. I guess the one thing to caution, maybe there's two cautions. One is many of these gene families have reached an optimum. It's not like just increasing copy number is always a good thing. And so I can think of an example like defensins, which are very variable and they reach certain levels. When they get too high, they predispose to psoriasis and problems with sepsis in humans, right? So too much is not always good enough or a good thing. So there's an optimum. So you can think of these like accordions where they kind of set, maybe adapting to the environments, and they can expand and contract at certain levels. The other caveat, and this is an important point, increased copy doesn't always mean more expression. And so you can have many copies of a gene and still have similar levels of expression to someone who has reduced levels, but you just have more genetic copies of it. And so a real classic example of that is like the red-green cone color pigment genes that allow us to detect wavelength. Men on their X chromosome have large numbers of that copy number or variation, but it turns out it's only the gene that's most proximal to the promoter and its regulatory sequence, which actually is expressed. And so you can have 5 copies and still your first copy be a dud, and it doesn't have any advantage to having those extra copies. So it's a complex relationship, I would say, but there are clear examples of increased fitness, in my mind at least, with increased copy.
Okay, thanks so much. And sadly, this will be our last question. It's going to be for Sarah Tishkoff, just because we're almost at time here. Sarah, I'm just going to read this one because I want to say it exactly right, and this is not my field. I'm a biochemist. Okay, so you suggest that khoisan are not rooted at the base of the African tree, but what about the old khoisan skeleton from an individual without introgression with the Bantu? It seems to suggest a very deep root, almost half as far back as the Neanderthal branch from our own lineage.
I think they're referring to a study of ancient DNA from a sample from South Africa, I believe, that was probably about 2,000, 2,500, I think, years old. And the idea was because there's no introgression or no admixture with Bantu populations at that time, that maybe it gives you a better sense of what the true evolutionary history was. And they inferred this very ancient time to most recent common ancestry. And other studies have shown that as well. And I think there's a couple of things that I note. One is that I do believe that when you study enough of the modern human genomes, the particular ones we studied have very limited amounts of admixture with Bantu. And they're very informative when you have good quality, high coverage modern genomes and you're looking across individuals across the genome. I think there's still a lot of power there. But the other critical thing is that we used modeling approaches that took into account things like admixture or migration between populations and changes in the population size going back in time, and also looking at inferred population divergence. It's only when you consider these more complex models that you see the rainforest hunter-gatherers and the khoisan being a sister clade that we think descended from a population ancestral to all other modern humans. And I think then it's showing you that not only is the data important, or the type of genomic information that we gather, it's how you analyze it. It's developing better computational approaches that can infer more complex histories. And I'll also be honest, I think that we still haven't captured it. I think the demographic history of Africa is so complex. There's a lot more to be known. And different data sets might show you slightly different things. So I can't wait to hear more about what others discover.
Great, thank you, Sarah. On that hopeful and forward-looking note, it's a wrap. Thank you all. Incredibly interesting talks and the discussion to follow. The breadth of disciplines that we've just bounced back and forth from is just amazing. There's nothing like CARTA. I'm a rookie at CARTA. I'm now co-director of CARTA. This is my first CARTA event, and really enjoyed being with all of you. Just want to thank all of our incredible speakers, for their very informative presentations. My co-chair, Katerina Semendafiri, On behalf of all of CARTA and the CARTA leadership team, I want to thank all our special supporters that made this symposium possible, the CARTA patrons, Ingrid Bernerska-Perkins and Gordon Perkins, Don and Natalie Handelman, Elizabeth Lancaster and Eli Schefter, our CARTA friends, John Starling, Donald and Carolyn Stewart, our CARTA supporters, Mary Fitz, Karen Matthews, Christopher Rodig. Thank you so much. Without your support, we could not have had this event. We also could not have had this event without our incredible CARTA staff that organized this, pulled it all together, our colleagues at the San Diego Supercomputer Center, the remarkably professional UCTV team. There were over 300 registrants for this event today. Not bad. And I can just say that, this symposium, like all past symposia, will live online on the CARTA link from the CARTA website. The CARTA past symposium have had over 40 million online views. So it's the gift that keeps on giving. Thank you again, speakers and participants. So that's it for this time, but we're looking forward to joining you next time with the next CARTA event, which will also be an online symposium on March 3rd, 2023. I think you got a taste of this from Terry Sinofsky's talk today. The topic for the March 3rd online symposium is going to be artificial intelligence and anthropogeny. And then I'm happy to say we come back in person for the first time, back in person on May 19th, 2023, in the auditorium. That's a picture of the auditorium at the Salk Institute. Salk and UCSD are, of course, co-partners in CARTA. Where the title of that event will be The Role of Myth in Anthropogeny. So, stay tuned for what comes next. Check out the CARTA website. You can all go back and review some of what you heard today, as well as previous symposia. We're going to see you next time.